Hello, my name is Wendy Zi. We are going to demonstrate the technique for both aspirating spinal fluid and injecting both study agent and sterile flush using a single sterile connection technique. This is the same one used for sterile intraventricular injection of antibiotics. This is the injection and aspiration apparatus. It consists of 3 syringes, 2 stopcocks, and 1 screw port on the end. The study drug is attached to the stopcock closest to the screw port. The flush is attached to the next stopcock, and an empty syringe for aspiration of spinal fluid is attached to the back stopcock. The apparatus screws directly into the port of the ventricular system closest to the head. Some hospitals may still use a needle port for injection. In this case, use a 25-gauge needle to puncture the port. Many hospitals use a needleless entry system, such as this. In this case, use a screw-top syringe. Current procedures for preparing products for intrathecal administration in the United States ensure that only the contents of the syringe containing study agent and flush are sterile. The outside of the syringes have non-sterile labels and come in non-sterile bags. When bringing the syringes into the sterile field with the 3-way stopcock apparatus, care should be taken to ensure the exterior of the syringes do not touch the IVC port or gloves that may touch the port. This may require a change of sterile gloves. The goal in this regard is to ensure the sterility of the fluid and fluid pathways to the ventricular system. I'm next going to prepare the ventricular drain with an antiseptic solution. I'm using a Providone iodine solution, which is generally used as the standard of hub care, although alcohol is also acceptable. You notice that the stopcock is suspended in mid-air, and we do that simply by hanging the tubing over the pole, and we prepare this as best as we can with the iodine solution. You notice just below the rubber port is a 3-way stopcock. In our system, the portion of the stopcock that moves in this direction is towards the patient, and the tubing from the stopcock going in this direction is towards the transducer. Because the tubing is clamped with these slides, whether we inject or aspirate, no fluid will move through this limb of the tubing, and the only route is for fluid to go into or out of the patient. If your system allows, it is highly recommended to monitor ICP during the injection by opening the system to the transducer while keeping the drainage bag closed. Once we've adequately prepared the port, I like to take the port with an alcohol rub or gauze, and then I can take the ejection apparatus and access the port like this. You'll notice that at this time, the stopcock containing study drug is off to the drug, the stopcock with the flush is off to the flush, and the stopcock for injection is off to the transducer. Now you see how with a single connection we're going to be able to use a 10 cc syringe to gently aspirate spinal fluid, allowing us to perform an isovolemic injection. And in fact, if you need spinal fluid for laboratory tests or for study samples, you can use this aspirated CSF for that sample. Now that we have that, we are going to turn this stopcock so that when I inject drug, the drug will go directly into the patient. So I gently inject the drug like this. Now that the study drug is injected, I close that stopcock again and turn the other stopcock so that I can inject the 3 milliliters of flush, like so. And this allows us to be certain that all of the study drug actually enters the ventricular system. And with that, we are done with the procedure. Although it may seem reasonable to re-inject the CSF that was removed instead of flush, we recommend using sterile saline flush in order to avoid returning blood breakdown products to the patient which may have adverse effects. We unscrew the injection apparatus from the port and reattach the plastic hub.
Okay, just pause.
If necessary, we can use this syringe of spinal fluid to send for CSF analysis. At this point, we open the port, open the 3 slides, and ensure that this port is open to the transducer and closed to the drainage bag. This allows us to monitor intracranial pressure for the 1 hour that the system is closed to drainage. And that's it.